Hello, in this video we will discuss about the respiration overview and glycolytic pathway. So what is the glycolysis, and that is the initial step for the respiration. So the series of enzyme control oxidation reduction reactions during which carbohydrates produces during photosynthesis. So the carbohydrate will be produced from the photosynthesis we will discuss first. So the overview of the carbohydrate production in the chloroplast, which that's contain a thylakoid membrane. Thylakoid membrane contain chlorophyll pigment. This chlorophyll pigment is used for the light to in the presence of a water to produce the oxygen, while the carbon dioxide used in the dark reaction to produce in the Calvin cycle to produce the glucose in the form of triose. So the glucose formation will be occur after two triose formation. So in this way, here is the glucose. This is the C6H12O6, and this oxygen is used during the respiration. So the when we will consume these things, so the glucose will be break down in the presence of oxygen to produce 38 ADP to convert 38 ATP, and a carbon dioxide will be produced after the inhalation of oxygen to exhalation of carbon dioxide in our body. So here the mechanism of respiration is the glycolysis to Krebs cycle, and the Krebs cycle is used also for beta oxidation of the fatty acid and amino acid used for it to convert acetyl CoA to produce energy. So in this way, the electron transport chain and ATP will be produced. This ATP uses stored and dead. So here the respiration is two type, aerobic respiration and anaerobic respiration. Aerobic respiration use oxygen. So in this way the electron transport chain is used for the oxygen and the Krebs cycle. So this is the all thing is the aerobic respiration, while anaerobic respiration is absence of oxygen to breakdown of glucose to produce alcohol in the form of bacteria, some yeast, or on the other hand you can see that is basically alcoholic fermentation and lactic acid fermentation due to the excess of exercise. So here is a respiratory question we will discuss now. Is the ratio of the moles of carbon dioxide released by a tissue over a period of time to the moles of oxygen taken up? Thus, RQ is the respiratory quotient value, is equal to moles of carbon dioxide released our mole of oxygen taken up. So in this way, the plant is also use the respiration for produce the energies, same like a human, same like an animal, because it also contain mitochondria and use oxygen also during the night time, and you should remember about that. But in the case of daytime, it's produce the oxygen, but nighttime it will be inhibit. If the glucose is a respiratory substrate, for example, substrate is for use the enzymatically activity to produce the oxygen, etc. Carbon dioxide and water, so also energy. So the RQ would be RQ is equal to 6 mole of carbon dioxide or 6 mole of oxygen. So in this way, the one it means the carbon dioxide also contains 6 molecules while the oxygen also contain, but in RQ value is 6 of the glucose, 1 of the glucose, sorry. So the fats are poor oxygenase, these are oxygen, these are the not electron directly oxidized. So the oxygen here is a trivalent fat is 6 oxygen, while the carbon dioxide is basically oxygen is 6 and 80 molecule of oxygen used to produce the 57 carbon dioxide molecule. So the 0.7 RQ value. So let's begin to understand. First step is the glycolytic pathway, because I have made a many video about the electron transport chain. You can watch that video in my channel. So here is, you can see this is the starch. So remember about that starch is synthesis in the cytoplasm of the plant cell. So here is the surrounded is known as the cell wall made up of cellulose. So in the cytoplasm, after the production of sucrose, for example, in the sugar cane, this is a composition of the fructose and glucose via a glycosidic bond after the condensation reaction to release water. But in this way, invertase enzyme used to release this glycosidic bond in the presence of water. So the addition of water to fructose and glucose separation will be occur And in this way, the glucose and fructose, under the condition of anaerobically in the cytoplasm without using oxygen, is known as the glycolysis. Glyco means the glucose and lysis means the breakdown. So after the separation, here is the starch we will discuss first. So this step is known as the glycolysis. This glycolysis is the starch breakdown. And in this way, after the production of starch, which that is with the passage of time, this will be used for the seed growth, etc., and the shoot growth, root growth. So here is the phosphorylation will be occur, addition of phosphate group in organic phosphate group. In this way also this glucose will be converted by a phosphorylase enzyme to produce glucose 1-phosphate. So this glucose 1-phosphate also convert by the isomerization. So this is a phosphoglucomutase enzyme to create a mutation. In this way, the glucose 6-phosphate conversion will be occur, and in this way, the phosphofructokinase enzyme use also by the using of ATP to produce ADP, and this is the basically the glucose 6-phosphate, the phosphofructokinase enzyme. This phosphofructokinase enzyme used to fructose 1,6-bisphosphate, 1,6-bisphosphate formation. So in this way, here is the phosphate group attached with the carbon number 1 and carbon number 6, and in this way, here is the triose phosphate isomerase enzyme used for production of these things. So in this way, important thing is that is the 3-phosphoglyceraldehyde and 3-phosphoglyceraldehyde will be formed, and 2 type of sugar molecule. This is the triose sugar, and this triose phosphate isomerase enzyme used to conversion the triglyceraldehyde and triacylglyceraldehyde, which that is basically converted by the addition of phosphate group, and in this way NAD plus will convert into the NADH, and in this way the 1,3-bisphosphoglyceraldehyde formation will be occur. Anyhow, here is you can see this is the ADP will use to produce the ATP, and to the phosphoglycerate kinase enzyme used to phosphoglyceraldehyde formation. This phosphoglyceraldehyde formation is basically here you can see this is the phosphoglyceraldehyde formation, and in this way the 3 carbon containing this molecule will convert into the phosphoglycerate mutase enzyme to cause mutation to create the 2-phosphoglycerate formation, and this 2-phosphoglycerate formation ultimately convert into the phosphoenol pyruvate in the presence of enolase enzyme, and this phosphoenol pyruvate ultimately convert in the presence of phosphoenol pyruvate kinase enzyme, pyruvate kinase enzyme used for it to produce the ATP also. So in this way you can see, and to produce pyruvate. This pyruvate is the result of anaerobic respiration to produce NADH and ATP. So the energy yielding process. This is the glycolysis. And after this, the pyruvate will go into the mitochondria or fermentation. So when fermentation will be occurring in the absence of oxygen, to can be alcoholic fermentation or lactic acid fermentation or aerobic respiration, aerobically fermentation. So this is the mitochondria and Krebs cycle, electron transport chain, and ATP. So in this way you can see and understand as well. But in the case of important things I want to share with you, this is the evolutionary processes. This evolutionary process this is the initial phase for the respiration to produce ATP, while the prokaryote, this is the prokaryotic lack of the mitochondria in the prokaryotic bacteria. So the bacteria contain just cytoplasm but not mitochondria. So in this way, if not mitochondria are present, so it means the oxygen is not using in this manner, and in this way in prokaryote the bacteria is the microorganism, previous living organism produces ATP by glycolysis only due to the lack of mitochondria, and yeast produces alcohol in the presence of carbon dioxide and absence of the oxygen. So in this manner, you should know about that. The evolutionary process begin the initial step of the glycolysis to produce ATP. So thanks.